Some technical issues. Now we are okay. We are on board, and I'm very happy to have Samer Hattar here, an old friend. He's a chronobiologist and a leader in the field of non-image forming photo reception. He's the chief of the section on light and circadian rhythm at the National Institute of Mental Health, which is part of the NIH. He was previously an associate professor in the Department of Neuroscience and Department of Biology at Johns Hopkins University in Baltimore, and is very known for his investigation and the numerous discoveries regarding the role of melanopsin and the IPRGCs for the entrainment of circadian rhythms. He has won numerous awards like Albert Leningrad Research Award and Sloan Research Fellow and Lucille and David Packard Foundation Fellowship. So the title of the talk will be Retinal and Brain Circuits Underlying the Effect of Light on Behavior. So that's all. Say hello and thank you very much to Samer. We are very happy to have you here.
Yeah, thank you, Sharon and Yossi, for the kind invitation. I'm really looking forward to an interactive talk. Please stop me if you can. And, you know, I'm going to stop every now and then and ask if there are any questions, just to make sure that everybody's following the presentation. Okay. Maybe if you move the first slide, Yossi, because then it will, yeah, just the first slide. Yeah. Okay. I lost control. Wait 1 second. It's not working from my end. Do you see my pointer? Yeah, you still see my pointer, right?
Yes, we do.
Yeah. Yossi, can you go to the second slide? My controls with the mouse are not working. Yeah, perfect. So as Yossi said, I started my independent career at Johns Hopkins University, and we were part of the Mouse Tri Lab, where myself, Hai-Chin Zhao, and Reggie Corvilla, 3 mouse neuroscientists, shared resources, reagents, and students and ideas. And for young faculty, I always like to present this slide because I think it's a brilliant idea for young people to feel they are part of a bigger lab. And in this environment, we were in the undergraduate campus, so we had more graduate students than postdoc. But next slide, please, Yossi. But in 2017, I moved to the National Institutes of Health, and this is not the most updated slide. I have to update that. The lab changed from a undergraduate lab to a fully, maybe mostly postdoctoral lab with some really amazing graduate students and postdocs. And I'm gonna talk about the work of people as I'm going through my presentation. So the rotation of the Earth around its axis has produced a very predictable and massively variable amount of intensity that occurs in the world. So for example, if you take 1 centimeter square of your skin in a sunny day at noon, you can get 10 to the 16 photons per centimeter square per second to hit that 1 centimeter square. And the same 1 centimeter square at night without a moon, you can have literally few photons. So this incredible change in the photon flux across the day and night produces challenges for vision of how could you see in this bright conditions and the dim conditions. But it was very beneficial for the animals to know the difference between day and night and to produce an internal timekeeping mechanism that allows them to anticipate these changes in the light environment. And one of the first organisms to do that was cyanobacteria, which use the light energy to do photosynthesis and the circadian clock to separate the photosynthetic pathway from the nitrogen fixation pathway. And this allowed the cyanobacteria to change the oxygen concentration in the air, which actually led to our evolution. So we could actually thank the daily rhythms and the light-dark cycle for our existence and our ability to see this presentation. So the mammalian eye is the main light detector in the body, with the retina as the light sensitive tissue of the back of the eye. And the eye is part of the central nervous system, but it buds off and is connected now to the brain via the optic nerve, which is formed from the retinal ganglion cell axons. And we're all familiar of the eye because of the conscious perception of images. So for example, to see this beautiful image here, light is reflected from this image, goes through the pupil, hit the back of the retina, and then goes to thalamic and superior colliculus and other areas to allow you to form a conscious perception of this image and also track it. However, the eye detects light for functions that are not concerned with image function and tracking, and these are collectively known as non-image forming visual function. Yossi, can you go to the next slide, please? And these include the very reflexive and very simple behavior, which is the pupil constriction and circadian photoentrainment. I would like to say that although the pupil constriction is very simple, it actually requires a very complicated circuit. So although you could imagine driving the pupil by simply putting a photopigment in the iris muscle and allow it to drive locally, and in fact, some animals do that, in the mammalian system, the pupil constriction requires a very complicated circuit where light has to go through the pupil, hit the retina, goes to an area in the brain called the auricular pretectal nucleus, and again through multiple synapses, come back again to the ciliary muscle to drive pupil constriction. And circadian photoentrainment, which is allowing us to have the sleep-wake cycle here. The circadian rhythms are driven by a region in the brain known as the suprachiasmatic nucleus, and I'll be telling you more about this later in my talk. So why should we care about circadian rhythm? As you know, we do have sleep-wake rhythms. As isotherms, we also have beautiful temperature rhythms. So it's really surprising to me when I went to the circadian field to saw that our body temperature changes within 1 degree between day and night. And although there is a correlation between sleep and temperature, they can be dissociated. In fact, sleep and body temperature dissociate when you have jet lag. And that's partially why you experience the symptoms of jet lag. And finally, what really shocked me is that even your ability to do cognitive functions varies across the day-night cycle. So I always used to tell my students when I was teaching them at Hopkins that although every person in the room is different, unless you have an identical twins, you as a single individual, you're not the same person in the morning, at early noon or early evening hours. You not only change physiologically, functionally, molecularly, hormonally, and even cognitively. So how do we then model this very complicated system in the lab? Well, thanks to the circadian field, we have really beautiful measures to accurately and very quantitatively measure circadian rhythms in the lab. And they are very easy. You could do it in any lab. You simply put a mouse in a cage and with a wheel. And every time the wheel moves, you can record the wheel revolutions. And then you put the mice under different light conditions. So here's, for example, 32 days, where in the first 13 days, we put the mice under light dark conditions, and we double plot the data so you could see the consecutive cycle on the same line. And then you put the mouse in constant conditions, constant dark conditions from day 13 to day 32. And what you see very beautifully is that when the animals are entrained, they have exactly 24-hour period. And because mice are nocturnal, their activity are confined to the dark portion of the cycle. And if you actually look at this very small activities here, these are like 300 wheel running revolutions per 10 minutes. So if you calculate the amount of running that a mice does in a single night, people suspect that it comes out to be 4 to 6 miles of us running on a treadmill. So what you also notice is that the circadian period in constant condition is not exactly 24 hours. Hence the name circadian. Circa mean approximate and day. So if you just hit the next slide, please, you'll see what you see is that if you see the red line, what you see is that although it's very precise, it's not accurate. It's actually really beautifully precise because it happens within few minutes of variation between day and night. And if you calculate the period length from this mouse, it's approximately 23.6 hours. You could jet lag this mouse. You could bring them from New York to Israel and Israel back to New York. And, if I had come in person to give this talk, I would have like at least 6 to 7 days to adjust to the new shift in the light dark cycle. And what you could see, this happens in both direction. It happens when you go to a country where it has an earlier sunrise, and it happens when you go back to your own country where you have a later sunrise. So what's really amazing about this system is that it's uninvasive, individual mice, very easy to record, but you could get very quantitative understanding of what is the sensitivity of the circadian system to light, what kind of light color you can do to entrain, and what is the intrinsic period of the circadian clock. So because light is important, people have assumed that it has to be the rods and cones that are involved. And the mammalian retina is interesting in that it's an inverted retina, which means if you take the pupil and you look through the eye, the first layer you see are the retinal ganglion cells, which actually form the optic nerve. Light has to cross all these retinal neurons before it hits the photosensitive parts of rods and cones, which contain the photopigment, rhodopsin and conopsins. These rhodopsin and conopsins are able to absorb a single photon and through a phototransduction mechanism, transfer this photon energy to an electrical signal, which they then send through the same parts of the retina that light passed through to the ganglion cells to send it to the brain. And it was really, honestly, the human researchers like Bud Brainerd here in the Stoke and Chuck Seisler and others that have started to implicate other photopigment in the mammalian retina that is distinct from rods and cones. But it was really the work of Russell Foster that convinced maybe the field at a bigger level when Russell Foster's group published 2 papers back-to-back in Science. And what they found is that animals that completely lack rod and cone through genetic deletions were still able to photoentrain similar to wild-type animals. That's quite remarkable because that said that there are other photopigments, photoreceptors in the mammalian retina that are able to signal light information to the brain. And to make a long story short, it was really the pioneering work of Ignacio Provencio and David Burson, and then in collaboration with us when I was a postdoc at Hopkins, that we discovered that a subset of ganglion cells are intrinsically photosensitive. That's a fancy way to say they are photoreceptor because they express a photopigment melanopsin, which is similar to the rhodopsin and conopsins that I told you about before. And so now we call these new cells we call these new things Ignacio Provencio retinal, intrinsically photosensitive retinal ganglion cells, and I'm going to refer to them as IPRGC throughout my talk. So because the experiment was so beautiful, I'm going to take some time of my work and just show you how David Burson have first discovered that there are photoreceptors outside the rods and cones after he saw the discovery from Ignacio Provencio that there are melanopsin expressed in retinal ganglion cells in mammalian retina. So we knew from the 70s that a very small subset of ganglion cells veer from the optic tract and directly target the suprachiasmatic nucleus, which I told you is the central location of the circadian clock. So David postulated that maybe in rodless, coneless animals, it's these ganglion cells that project to the SCN that send the light information. So what he did, he injected the rhodamine dye in the SCN, and then he looked at the retina, as you could see in the retinal flat image. Yossi, can you go one more click? Yeah, and he was able to label very few cells, as you would expect. If you injected dye in the SC or the LGN, you label a lot of ganglion cells, but David only labeled between 25 to 40 ganglion cells. So then what he did, I think, was brilliant. He blocked all signaling from rods and cones to ganglion cells, and he recorded from on ganglion cells, the one that respond to the onset of light. And what he found is if you block the rod and cone signaling and record from ganglion cells that are not labeled from the SCN, next slide, what you see is that there are no responses, which is not surprising. Ganglion cells are not photoreceptors. If you block rod and cone from signaling the information, they'll be completely unresponsive to the pulse of light. However, when he shined light on the cells that project to the SCN, what he found was remarkable. Not only these cells responded to light, but they depolarized. By the way, rods and cones hyperpolarized to light, and they fired action potential, implicating that they send this information directly from the retina to the brain. And what was really remarkable is that David found that all these cells that project to the SCN belong to a ganglion cells that have their dendrites in the off sublamina of the retina, which usually these ganglion cells receive decrement in light intensity. But although now we know that they have their dendrites in the off sublamina, we know that they are both light responsive and receive on input. And I'll be happy to answer questions at the end about what I think is happening here. Next. So because these IPRGCs project directly to the brain, I wanted to see where they project in the brain. So to do that, we replaced the melanopsin gene with the tau-like-Z marker gene, which allowed us to label both the cell bodies and the axons of these IPRGCs, as you could see in the retina image. And what was really remarkable, because this is a chromatic staining, you could see very beautiful that there is very little background. So you could now take the axons that go from the optic disc and see where they project in the brain. And here's a ventral view of the whole brain. Here's the optic nerve. Here's where the eyes would be located. And you could see very nicely that there is a very beautiful innervation from these IPRGCs to the suprachiasmatic nucleus, the central location of the circadian clock. So we call these ganglion cells circadian photoreceptors to quickly actually notice that the SCN is only one of many, many regions that these cells project to. And in fact, now we know that these cells project, in addition to the suprachiasmatic nucleus, pupil centers, sleep and alertness center, and mood regulating centers. And this talk, I'm going to tell you a lot of new information about IPRGC regulation for sleep and mood regulation functions. So just for completion, really, maybe like literally 6 or 8 years after the discovery of IPRGCs, we thought that they are only comprising one population, which is the M1 population that I told you about earlier. But work from my lab and others from these 3 amazing women have shown that IPRGCs are at least 5, and now new data from David Berson's lab says there's even a 6th form of IPRGCs, and that they can be easily differentiated on very specific morphological and electrophysiological properties. And Tiffany Schmidt in her own lab has done beautiful studies with M4, which are the on alpha on ganglion cells. We know that the M4 are actually the famous contrast detecting on alpha ganglion cells. But regardless of the complexity of the IPRGC population, we wanted to ask a simple question after discovering melanopsin and IPRGCs. Are rod, cone, and IPRGCs the only photoreceptors in the mammalian retina? And this wasn't a trivial question at the time because there was a lot of research that implicated cryptochrome, a flavin-based photoreceptors, as the circadian photoreceptors for the mammalian circadian clock. So to answer this question, we produced triple knockout animals where we destroyed the phototransduction pathway of rods, cones, and IPRGCs. And when we did this, now you could see that the animal, although have normal eyes looking, although the eyes connect to the brain, although there is no degeneration of rods and cones, they completely are incapable of entraining to the light-dark cycle. In fact, you see that these animals completely free run throughout the light-dark cycle. I just want to remind you that rodless, coneless animal and melanopsin knockout animals photoentrain in a similar fashion to the wild-type animals. Although, again, I don't have the time to show you, we think melanopsin knockout animal does have deficit, but they are not as pronounced as the triple knockout animal, as you see here. So the question then we wanted to ask, are IPRGCs then, since melanopsin is not necessary for photoentrainment, we wanted to ask, are IPRGCs the only relay for light information to reach the circadian clock? Or could it be that other ganglion cells can also contribute to circadian photoentrainment? So to do this, these 2 amazing people in the lab, Ali Guler and Jen Ecker, decided to use the attenuated diphtheria toxin, which attenuated diphtheria toxin A subunit, which specifically inhibit translation. And so what they did, they expressed this toxin under the melanopsin promoter, so the hope that this will kill all IPRGCs. But actually what we found is really very lucky for us, to be honest. Because we used the attenuated diphtheria toxin, we found that we only were able to ablate the M1 IPRGCs because they have the highest expression of melanopsin. We didn't touch the non-M1 iPRGCs, and remarkably, even the M1 iPRGCs died only at adult stage. And because of that, image-forming vision and development were not affected at all. So we actually produced an opposite animal to the animal that Russell Foster produced, in that Russell Foster animal is image blind, but able to see circadian photoentrainment. We now have an animal that is image capable, and we wanted to see what happens to the ability of the animal to detect a circadian photoentrainment. And we honestly were stunned, because this animal looked exactly like the triple knockout animals. And to me, that's just a remarkable evolutionary feat, to think something as important as circadian photoentrainment will require only a very small population of iPRGCs to photoentrain the animal. And I always tell my student at Hopkins again, is that this is like having a mathematician who's she's so smart, she could solve any mathematical equation, but then you ask her, what is 1 + 1 = 2? And she cannot tell you it's 2. Because, you know, it's these animals still form complex image formation, but they are not capable to tell when the light is on and when the light is off. So, so now we know that the circadian field has always thought that the eye projects to the SCN, which has a central pacemaker, and then that regulates all your peripheral clock. And now we know that this RHT, or retinal hypothalamic tract, is dependent fully, next slide please, Yossi, on, at least in mouse, a single population of iPRGCs. But when I moved to my NIMH, it was really great that we continue to work on the SCN and all the beautiful mechanism that the SCN do to entrain the whole animal. But I wanted to ask a different question. Does light directly influences behavior? And this question didn't come out of nowhere. It came out based on the morphological studies that I showed you before. Because as I showed you, iPRGCs project to many different regions that are distinct from iPRGCs. So I'm going to give you a summary, and then I'm going to show you a specific data point to show you how interesting the system is. So next slide. So a summary of how we did these results is, you had to produce an animal that somehow keeps the SCN innervation intact, somehow keeps all conventional ganglion cells intact, and only remove the non-SCN projecting iPRGCs. And again, thanks to Alan Chen and Melissa Simmons, not shown here, we have produced this animal in using intersectional genetics between the melanopsin Cre line and Brain3B DTA line that only express DTA in the presence of Cre. So in this way, only cells that express Brain3B transcription factor and melanopsin will be killed. And to give you the summary first, and then I'll show you the data, we found that all SCN projecting iPRGCs are intact in these animals. All the visual center projecting RGCs, apart from the M4, are also intact in the animals. But if you look at the middle of the brain, these are the non-SCN projecting fibers, and you could see that they are completely eliminated in the Brain3B animals. So the OPN, the other brain regions that, yes, the OPN and the other brain regions that melanopsin project to was completely denervated, but the SCN was completely intact. And consistent with this, we found that these animals, since they have complete innervation of the SCN, are fully capable of photoentrainment. So although we killed nearly 90% of all iPRGCs, and we only had 200 M1 cells in each retina, these animals photoentrain. They have normal vision under full contrast, because the M4s are important for contrast. So they have normal vision, they have photoentrainment. So we could use these animals to differentiate between the SCN circadian pacemaker pathway versus the non-SCN circadian pacemaker pathway. And now let me show you what we discovered, and then I'll go through some, and I'm happy to answer questions about others. We I think made 5 major discoveries with this system. The first, which I'm going to talk a lot about, is the mood control center and the learning centers. I'm going to talk more about this later. And I'm going to talk about sleep control with the preoptic area. We also found that darkness, if you silence iPRGCs, can influence alertness in mice. And a very interesting study done by Diego Fernandez also showed that IP-RGC input to the SCN can affect time-restricted feeding. I won't have time to tell you about alertness and cognitive functions and feeding, but I'm going to tell you a lot about mood and sleep control. So how first we could test whether light can directly affect behavior independent of the circadian clock. So when I started my lab, I wanted to split from my postdoc advisor. So I wanted to come up with some new ideas to work in my lab. And I came up with the ideas that maybe light can affect behavior independent of the SCN circadian pacemaker. It's easy to come up with this idea, but it's very hard to test it. Why? Because I was telling you the whole time that light affects circadian rhythms and sleep. So how could you test the direct effect of light on behavior independent of disrupting circadian rhythms and sleep? So in my whole talk, I've told you about the T24 cycle, where we give 12 hours of light, 12 hours of darkness to the animal. But we also, next slide, two brilliant graduate students at the time, Cara Altemus and Tara Legates, came up with a cycle that has been known in the circadian field, but they modified it in a way that they can give this T7 cycle, 3.5 hours of light, 3.5 hours of dark for 2 weeks. And that when they do that, this cycle allows them to present light at all circadian phases without causing sleep deprivation and without causing circadian rhythmicity. Next. And when they did this, and they just put the animals under either the T24 or the T7 cycle, what they found is quite remarkable. So here are animals placed under T24 and animals placed under T7. They are from the same vendor. They are wild-type animals. And they use the behavior that mimics despair in a way in a human, which is observed. Despair usually is observed in patients with depression. So this test is not the best test for depression-like behavior, but at the time people were using it extensively. And the amount of immobility, so this is where you put a mouse in a beaker with no ability to escape. And the amount of immobility is supposedly correlated with depression-like behavior. And what you could see that there is a much higher immobility in the T7 animal, indicative of a higher level of depression-like behavior. But in the next behavior, which I really like a lot, is more a craving behavior. In humans, depression is more complex. Some people crave more food, some people crave less food. But in mice, it seems that if you have depression-like behavior, they actually have less preference for sweet stuff. So this is called the sucrose preference test, where you give an animal 2 water bottles, one spiked with sucrose, and usually animals maintained under normal light conditions drink between 70 to 80% of the time from the sucrose spiked water, but animals under the T7 cycle indicative of depression-like behavior drank much less from the sucrose spiked water compared to the normal water. In addition, we know that depression is usually associated with some learning difficulties. So we did a learning and memory test, which depends on the hippocampus and this short-term memory. This is called the novel object recognition test, where we put a mouse in a environment with 2 familiar objects, and then we took them 1 hour out, and then put them in the same environment, but now we gave them a novel object. And mice are just like us, inquisitive. I mean, you know what happens when a new iPhone comes in. Everybody wants to play with the new iPhone, and they forgot about the old iPhone. And so that's what the mouse does in a way. It spends 7 minutes of the 10 minutes we recorded investigating the new novel object compared to the familiar object, if it remembered seeing the familiar object. But if it had learning and memory issues, then it will spend the same amount with the familiar and the novel object, as you see in the T7 cycle. Next. So this allowed us to really come up with a quite honestly bold conclusion at the time, which I kind of heard from people, that there has to be some sleep and circadian influence for many years after the publication, in that just by changing the light environment from T24 to T7, you can actually induce depression and learning and memory deficit in mice. And what's really remarkable to me, that if you use the animal that I told you we killed, the M1 IP-RGCs, the ADT animals, you actually block the negative aspect of light on these behaviors. And in the next slide, we then came up with a very nice model at the time. We said, look, we're not going to debate whether disrupting circadian rhythms or sleep can cause mood and learning and memory. There's tons of data to prove that. But what's really interesting to us is that light through IPRGC can have a direct effect on mood and learning and memory, independent of circadian and sleep problems. And remarkably, at least in rodents, I don't know if this is true in humans or not. I'm not so sure it could be true because we are much more visual. Image-forming pathways do not seem to contribute to the depression and learning and memory deficits that we observe. And I'm going to stop here for a second because this is more of a published stuff before I go into the new stuff and ask if anybody has any question. If there are no questions, I will continue.
I have a question, Samer.
Sure.
Are there any hypotheses on the direct path of this mood?
Oh, great question. I think then that's a great way to go to the second part. I mean, so understandably, we didn't figure out the brain regions, so there's still skepticism. But now I'm going to tell you about the brain regions that are involved in these behaviors. So next slide.
One moment.
So, yeah. Next. Yeah. So it was the work of Diego Fernandez that with Ruth Chikomal, who was not shown here, that did some of these studies. And what is really interesting is that if you now use the brain 3B animals that I told you have only the SCN innervation, and you use the sucrose preference test, what you notice is that, as shown before, you have higher level of depression in T7 control mice, but the SCN innervated animals really show no depressive-like behavior under the T7 cycle, even though circadian rhythms are still there and circadian photoentrainment is still there. So we were so surprised by this data that this is real number of experiments, and we had to make sure 100% that this is a true effect. What was even more surprising to us in the next slide is that when we looked at learning and memory, we again repeated what we observed in the control in the previous publications. But now when you look at the brain 3B animal in the lower panel, next slide. Yeah, what you see is that there is a deficit in learning and memory in these animals. So it's really interesting that although we didn't see a depression-like behavior, the SCN seemed to contribute to the cognitive function deficit. And in the next slide, what was really remarkable, this deficit in mood regulation was independent of the circadian pacemaker. So this is actually a rhythm of a circadian clock gene, PER2, that is measured in the SCN from T7 animals, and it has the same beautiful rhythm you observe in the T24 animals. So what we found then, next slide, is that remarkably, as to your question, Yossi, the direct pathway is much more complicated in that not only there is a direct pathway that affects mood and learning and memory, they actually do it through different population of IPRGCs. And they do it in a different need for the clock. So for example, surprisingly, the learning and memory which requires the SCN is clock independent. The clock is still functional, but you still get cognitive deficit. However, the PHB, which I haven't told you a lot about yet, but I'm going to tell you in a little bit, is actually required for mood regulation, but it is a clock-dependent function. So we actually found recently, which I didn't add the reference here in a Science Advances paper that we published, that this T7 cycle disrupts the only clock that is disrupted under the T7 cycle is the one in this perihabenular nucleus. So what is the PHB? The PHB is this really amazing region that we discovered that is beside the lateral habenula. Lateral habenula is the stress-induced, stress-responsible region for depression. And this really abuts the lateral habenula. That's why we called it the perihabenular nucleus. It receives direct input from brain 3B positive IPRGCs and then send this information to many brain regions that are very well known to be important for depression in both the humans and animals, and also reward and addiction. So these include the nucleus incumbens, the dorsal medial striatum, and the ventral medial prefrontal cortex, including the prelimbic and the infralimbic part of the prefrontal cortex. So that was really an amazing, to me, and if you go back, sorry, one slide, you'll see that was really an amazing discovery for me, because people have known for many years that people living in northern parts of the world experience depression-like behavior when the day length is shorter. And people have known that light can have positive impact on people with neuropsychiatric diseases. But because we didn't have an associated brain region, people really didn't believe these results very much. But I think now that we discovered that there is a real center in the brain that connect IPRGCs to the regions in the brain that are important for depression, makes us wonder if this, first of all, is found in us. We still don't know that. We only know that it's found in rodents. And whether that's the connection link between the depression that people observe in seasonality or the beneficial effect of light. So what is really interesting is that, as I told you, this region also projects to the prefrontal cortex. And the prefrontal cortex is famous because of this very well-known story of Phineas Gage, who was a railroad worker and had an accident where this motor rod in his hand went through his eye orbit and literally destroyed his prefrontal cortex. And the prefrontal cortex is really exuberant in humans. It grows very big compared to other organisms. It seems to control our conscious emotions, our social behaviors, our executive memory, and it's really divided into two major parts. It's divided into the dorsal part, which is more for cognitive, executive memory, and other behaviors, and the ventral part, which is the more ancient part, which is important for social and emotional behaviors. So the fact that the IPRGCs through the PHB in two synapses project to the emotional part of the prefrontal cortex, we wanted to ask a simple question: what happens if you denervate the PHB from IPRGC input? And the reason we wanted to do this is twofold. As I told you, this region receives input from the PHB and the layer 3 and 4 pyramidal neurons. And these layer 3 and 4 pyramidal neurons in the ventral medial prefrontal cortex are very sensitive to stress and circadian misalignment. So what happens when you have stress and circadian misalignment, their apical dendrites lose complexity as measured by the Scholl analysis. So Lorenzo Osprey here, a postdoc at the time in the lab, decided to actually do a very simple stain, which is the Golgi stain, and take neurons that are very isolated and look at their apical dendrites, which is sensitive to stress and circadian misalignment, and see what happens if you denervate IPRGCs. And let me show you the data because it's stunning. So first of all, if you have stress and circadian misalignment, you do get less complexity, but you don't get the same destruction of complexity that you see it when you denervate IPRGCs. And Lorenzo was very smart, so he actually did two different animals to test this. He did the ADT animal, which lack all M1 IPRGCs, lack the innervation to the PHB, but also lacks the innervation to the SCN. So it's not in training. So this could be a circadian effect. But you also use the brain 3B animal that I told you about, where the SCN is fully innervated, but the PHB is denervated. So these animals have normal circadian photoentrainment. And we know that both ADT and brain 3B animals under normal conditions, they don't have higher depression. So when he showed these changes in the apical dendrite complexity, we now know that these changes are not due to depression or circadian misalignment or stress, but it are really truly due to the lack of innervation of IPRGC to this PHB. Then he went a step further. He said, we know that ADT animals lose IPRGCs in adult. So what happens if you look at the apical dendrite complexity before the IPRGC loss and after the IPRGC loss? And you could see very beautifully here that the complexity is highly dependent on the presence or absence of IPRGCs. Then he went on and did a time course study. And he did a time course studies where he looked 7 weeks when IPRGCs are still nearly completely intact, 3 to 4 months when they just start to be lost, and more than 6 months, which we know most of M1 IPRGCs are lost. And you could see very beautifully in this schematic drawing in the right that the complexity of the apical dendrite is highly affected. So the fact that the complexity of the dendrites were affected, we wanted to ask, what about the function of the ventral medial prefrontal cortex? And the ventral medial prefrontal cortex, as I told you, is important for emotional control. So it really responds to anything that affects the emotion of the animals. So for example, if you get a very loud sound to the mouse and you record GCAM from the VMPFC, you could see a very strong response to that jarring sound. You could see that respond in the red here in the animals that lack IPRGC innervation to the PHB, but the response is much highly reduced, consistent with lower dendritic complexity. And I'm not going to show you the data. We found that this is also reflected at the molecular level, because of the lack of time, at the molecular level in the single cell sequencing from the ventral medial prefrontal cortex. Next. So as I told you, the ventral medial prefrontal cortex is important for social and emotional behaviors. So we wanted to see what happens in animals that lack innervation to the IPRGCs, to the PHB from the IPRGCs. And we did this free interaction test where if you use 2 males before sexual maturity, before they have mated with females, and you put a mouse in the cage and then you give an intruder mouse, what happens is that what you see in this blue line here below in the control, mice start investigating each other, then they spend a lot of time very close to each other's. However, what we found remarkably is that the IPRGC ablated animals not only don't want to investigate the intruder mouse, but they seem to be running away from this mouse. And I'm sorry that the video in the next slide is not going to work. But if this video works, what you would see that this mouse, the ADTA mouse that is in the corner, is running in its cage, and you put an intruder mouse and the mouse will be running away all the way in the cage from the intruder mouse, indicating that the deficit we've seen at the level of morphological, molecular, and responsiveness of the VMPFC are also reflected at the behavioral level. Next. So then we wanted to do something really interesting. So we know that the looming test is a visual test, and looming test is really remarkable. Actually, it's a beautiful test. So you give an expanding black disc to the mice. And if you've ever hiked and you got a very big, birds on top of you, you could actually see that shadow of the bird coming over you, and it's kind of scary. And so it's the same for the mouse here. When they see the expanding black disc, they think there is a prey, prey is swooping down to catch them. So what happens is that they either freeze or they shelter in the shelter that is given to them. And consistent with our previous data that the animals that lack, can you go back? Sorry. Yeah, one. Consistent with the data from previously that the ADT animals can form images, you could see that both wild type animals and ADT animals in red are sheltering and freezing to the same amount of light. Next, however, if you actually look at this behavior, there's something that Andy Huberman have discovered, that tail rattling. I wish this video plays. You could see that when the disc expands, the tail of the mouse starts rattling. People think that this tail rattling is a way that the mice communicate dangers to other mice. And this tail rattling is dependent on the medial prefrontal cortex. So in different from the visual cortex or the image forming centers that are dependent for sheltering and freezing, the tail rattling is dependent on the medial prefrontal cortex. And now when you look at tail rattling in these animals, next slide, what you see is that the control animals tail rattle at least 74% of the time, but only 26% of the time in the ADT animals. So in the same behavior, you could see normal visual functions that are non-MPFC dependent on other cortical and non-cortical region, but the MPFC dependent effect is highly attenuated, again consistent with the data that I've showed you. Finally, we wanted to use a test that is not dependent of the innervation of the IPRGCs, because one of the problem is that people thought that it may be a trophic effect that affects the complexity of these dendrites. So we then put the animals simply in constant dark conditions from birth. So we didn't do anything to these animals. We didn't kill any IPRGCs. We simply put them in constant darkness. And remarkably, what you could see, just putting the animals in constant darkness also affects the complexity of the medial prefrontal cortex. So to me, this really is mind-boggling, because it tells me that not only light is important for our mood and emotional well-being, mood and cognitive function, circadian rhythms, vision, but it also may be important for our social behavior. And if you actually think about it, it's not such surprising, because light is possibly the best organizing factor for any social behavior in animals. And to have that direct pathway from IPRGCs to the medial prefrontal cortex maybe allows social synchrony between different members of an organisms for, breeding, for better survival, and so on and so forth. And with that, I would like to thank both the funding agencies in the outside, especially the Packard Foundation, that helped me do all the mood and learning stuff when I started my lab and I didn't have grants to study them, as well as the very generous funding that I'm getting now in the intramural program. But I would like to specifically thank two important people here that helped me do the work. Tudor Badia, when he was in Jeremy Nathan's lab, who actually allowed us to come up with the brain 3B animals to do all these experiments that would not have happened without this animal. And Yogita Chudisama, who's really working very closely with us on the ventral medial prefrontal cortex, an area that I didn't know anything about before we discovered that IPRGC projects to, and without her expertise and her lab expertise, it would be very hard to do these studies. And with that, I'll be happy to take any questions if you have any.
Okay, Samer, thank you very much. This was really exciting. Let us all first thank you. Thank you very much. And yes, are there any questions? Please unmute yourself and ask a question.
I'd be delighted to start. So I understand that most of the work was not done on humans, and first of all, I'm interested to know what you think, how this would or would not generalize to humans, because there are differences between the rodent and the mammalian or the primate or the human visual system. That's one thing. I also want to ask about whether in any way this is related to the coniocellular mechanism, which is really kind of mysterious. I'm just wondering. And also I wanted to ask, I mean, I saw that you were talking about ambient light, but is there specific range of wavelengths that are more effective for these? Are they expanding more than, the typical light that is absorbed by the typical photoreceptors, or is it some I don't know about the ranges, and whether you could UV or ultraviolet, or is it some monochromatic range in the middle that would be effective for that would be like the common denominator for everything? So yeah, these are my initial questions.
Wow, there's a lot. Let me first start with the very simple one, the coniocellular pathway. I'm so excited about this. We don't have any data. I suspect it may be, but we don't have any data for it. So that you're right. Absolutely. That's an easy answer, because we don't have. I'll go to the wavelength next, because it seems it's the easier one, but we know it's more complicated. The IPRGCs themselves absorb maximally at 480, but all photoreceptors have a range of responses that follows a wide range of wavelength. But mostly, if you want to think of humans, it seems that more red light is less effective at affecting non-image forming functions than the blue light. That's the simple answer. There's a lot of complication about which type of cone contribute, what's rod's contribution. I myself struggle with these data still. Sometimes I'm so confident, sometimes I'm not that confident. So there is a lot there that I'll be happy to discuss with you on in person. But now I'll go to your first question, which I have a lot of information about. And I think it's really a fantastic question because the sensitivity and responses of each behavior can tell you a lot, even without knowing which IPRGCs. And we know for a fact that, for example, circadian photo entrainment is the most sensitive non-image function through IPRGCs. If you think of IPRGCs have the most sensitivity to circadian photo entrainment, which is very surprising to me. Pupil constriction, they have nearly the least photosensitivity. Pupil constriction, IPRGCs are required, but they depend mostly on rod cone input. For the cognitive and mood behaviors, we haven't done a good dose response curve like circadian photo entrainment and pupil constriction, but we think it's going to be at least as insensitive as the pupil constriction. So it's really remarkable to me because in nature, even though they use IPRGCs, the sensitivity seems to vary, tells you that the animal can actually use different environmental light intensity to affect different behavior. And it really is interesting to me because a lot of people in the human field just tell you that sunlight seems to be doing something more than lights in the laboratory. But the evidence is not there. Nobody has done the studies really very carefully. So it could be that the sunlight has much bigger effect because it affects these mood and cognitive functions in human. However, as your question beautifully said, we don't even know if these functions, really these behaviors, these regions exist in humans and primates. So the first thing, sorry, we're going to do quickly in my lab, hopefully, is work with marmosets first, and maybe even macaques, and for sure we're trying to see if we can detect this region in a human with MRI. Fantastic questions, very fast answers. Happy to expand.
Thank you very much. Thank you very much.
Yeah. George, would you like to take a question?
Yes, Samer, a brilliant talk, as always. And I'm just looking for clarifications on things that I may have missed what you said. The M1 IPRGCs are required for the effects of light on mood and behavior. There's no role of rods and cones that you're detecting at this point.
Man, I wish I could say that with certainty. We know rods and cones input to them. Yeah, I mean, that's why when I tell people, yeah, I mean, this is where we need a more interesting system like your system, but the humans where rods and cones are separated with wavelength. In the mice, they are so close, and knocking them out, affects circuits and stuff like that. So yeah, I'm going to be disappointing you and say, I think it's mostly a melanopsin based, but I cannot completely rule out rod and cone input.
That's fair enough. Did I also hear you say that for those effects, the SCN is not involved?
I did. And that was very bold claim before finding the PHB, at least for mood. And but now we know that for mood, the SCN is not involved. For cognitive function, though, the SCN is involved, but not the circadian pacemaking function of the SCN. So right now we're sequencing the SCN deeply, like most people, and we're trying to do genetic intersectional model to try to see if there are different population of the SCN that receive light for circadian photo entrainment versus modulating cognitive functions, and hopefully that will happen in a couple of years. Brilliant question.
Great talk.
Thank you.
Yeah, I have a question, Samer. So you say that if you disrupt genetically this IPRGC's circuits, then you get many effects on behavior. I would say, I would just hypothesize, or maybe there are plenty of data about it, that it also happens if you have, like, in a complete dark environment, you will probably see. But what are the dynamic of this? I mean, if you disrupt it, and if you could, just handle it and do it temporarily, what are the dynamic of getting? And the other question is, these are nocturnal animals, so why do they need this light stimuli to interact?
Okay, I mean, honestly, my answers, Yossi, is going to be very unsatisfying because I don't have a full answer. The first one, the dynamic, we didn't do a lot of studies where we restore or put down, because these experiments are very hard. Honestly, they are already a lot of work. But I'm happy to work with anybody who's interested in this. We'll give you all the details of the T7 cycle, any mouse model we have, because I think these are great questions and we haven't done any dynamics, so I can't tell you, unfortunately, anything about that. But for your second question, which I think is more disturbing and interesting question, is that why would a nocturnal animal need light? And, need light for a very important control of their social behavior. I think it goes down to the fact that, and this is a very hand-waving answer, is that nocturnality and diurnality is controlled by many factors, and it doesn't directly implicate the height of light. So the animals decide when to be active evolutionary because of either resources or lack of predators, but that doesn't mean that light is not doing a good function for them. So one thing that really shocked me, that I looked at the retina of blind mole rats, and I saw that although they have degeneration of rods and cones, they still have higher level of IPRGCs in their retina. So even these blind mole rats still didn't get rid of IPRGCs, implicating that they may use the light for something. I think mostly it's being used for circadian photoentrainment, but that's why, to Sharon's question, to your question, it would be great to see if these effects are observed in a diurnal animal like marmosets, and in a human, for sure. That would be, that would be the ultimate. These are initial studies, and they will only be completely solidified when we do find this region in diurnal animals. And we haven't yet, because we haven't looked very hard yet.
Sorry, funny enough, there is a kind of a ranking of happiness in the world that was published 2 days ago. And the first 3 countries are Nordic countries. And the fourth one, which country is the fourth one?
Which one?
Israel.
No, but it's from 3 years ago, so don't get the wrong impression about the current situation.
It's nothing to do with the political situation now, but it's funny that 3 of these countries, the first 3 have something to do with light, and they are the most happy countries in the world. Yes, so maybe too much light sometimes is also too, and maybe too little. It depends on the season.
Yeah, I think it's very interesting. Clearly, that the thing that I like, maybe I didn't make this very clear, is that this PHB is really abutting the lateral habenula, which is also a stress control. So I think clearly light is not going to be the only factor. That's why not everybody has seasonal affective disorder in the winter. I think if you combine, and that's what we're trying to do, we recently started working on stress in our lab. If you compare light and stress, you may even get more, either compensate or make stuff stronger. So I think it's really a complex system, and we're stripping it down to its essential elements. But you're right, if you start thinking about the humans, there's going to be a lot of other complicated factors to think about when you do that. And luckily, I'm working with Kathleen Merikangis, who works on bipolar patients, and hopefully we start making some progress there about the human function. So.
Oh, I was just going to ask you about bipolar people. They're very interesting in that they switch mood and they switch their behavior, and we don't really know. Have you got anywhere with that yet?
Honestly, not. The only thing is, I would say, is Kathleen's data showing that activity is possibly one of the biggest predictor of the switching, and especially afternoon activity. So it seems to be related to some either light or circadian function. Unfortunately, we have only one mouse model that is a bipolar model, which is the clock dominant mutation. And we don't have a lot of understanding of how this happens at the circuit level. But I think what is interesting to your question is that a lot of people are finding that surprising to me, but that's what the data is happening, that just blocking a very small part of the spectrum, the blue light blocking glasses, seem to be helping a lot of people with bipolar disease. So it's starting, some pictures starting to emerge, but it's a great question. And again, I'm sorry, it's not the most satisfying answer as well.
Thank you.
Um,
I don't know if we have time for a last question. Is it okay, Sharon?
yeah. I also have a question after you.
Okay. Sorry. So it was a really fascinating talk. I was wondering, given your comments about the periabenula and the findings of depression, if you think that it is specific to depression, or do you anticipate to find similar outcomes on anxiety? Or
I mean, we thought at the beginning to call it depression center, but now its effect on social behavior. And we published a recent paper in Science Advances showing that it's really complicated region. Although it's small region, it actually made of two different major cell types, an inhibitory and excitatory. The inhibitory ones also receive direct input from iPRGCs, which is mind-boggling. So again, I'm sorry, like all these are unsatisfying answer, but I don't think so. And I think there may be some even interesting interaction with the lateral habenula that we've yet to discover. I think there is a lot to do in this region. Yeah. The simple
Have you tested any anxiety models with?
Oh, that's a great question. Thank you for mentioning this. All the experiments we've done surprisingly didn't have an anxiety effect on the animals, at least not consistently, which is very different than from humans. Again, to Yossi's question, that depression and anxiety usually are comorbid. So clearly our studies are very important for circuits, but we have to have people like Bud, people like Kathleen, we have to carry them in a smart way to the human population, because otherwise we really could be misleading about the outcome. And not to mention that these behaviors only model a single aspect of the disease. They cannot model the complex aspect of depression in humans, right? Or anxiety. So these are great questions. Again, hopefully the future will allow us to do that.
Thank you.
Thank you for the question.
Samer, I have another question about, I mean, I know that people undergoing a depression due to lack of light sometimes are treated with phototherapy that is typically using ultraviolet light, or sometimes, and to the best of my knowledge, let's say.
I think that may be a bad idea to use ultraviolet, but because honestly, you don't need ultraviolet to activate iPRGCs, and even our blue cones, short wavelength cones, and hopefully Bud will correct me if I say something wrong, they don't need UV to be activated. So I would, considering that there are some, detrimental effect of UV light, I would just go with a normal bright white light. I think that should be more than sufficient in my mind, based on what we have. But again,
so it's not that I know the medical reasons why they reached this decision. Maybe it was just trial and error, but I was wondering if there is some reason to this specific light, the wavelength that that have been used. I mean, I remember that even as a child, but I know that people use it even in North America and stuff like that during winter times. People have
a longer UV light. They still also can activate melanopsin very well, because melanopsin sensitivity at the shorter wavelength doesn't go to 0. But I would just say, honestly, a blue 480 or even just a normal daylight white light, which is the safest. Yeah, I mean, if there is possibly the best, and it has to be diffused. I think it's important to be diffused because you don't want people to just activate the fovea. It's not like vision. You need to activate the whole retina, because iPRGCs are distributed all over the retina. So I would say UV is not necessary. Absolutely is not necessary. That's based on my data and the literature evidence, but I'd love to see the paper and discuss it offline with you, Sharon, anytime.
Yeah.
So.
Okay. Yeah. I don't want to take any more of your time, but if you do, I've got more questions.
No, I would love to Zoom with you and discuss it, because I could have missed something important, and sometimes you don't know. stuff can be weird in science.
No,
yeah.
it's fascinating. You haven't missed anything. It's just that my imagination about, I've got more questions, but no problem. Yeah.
Awesome. Yeah.
Okay, so if there are no other questions, thank you again for this really fascinating,
Thank you for the wonderful
enlightening talk, and let us all thank again Samer, and looking forward to see you in Arvo. Yes.
Yeah, I'll see you guys. Thank you so much. Bye.
Thank
Sharon,
you.
what is going on next week?
See you,
Next
everyone.
week, the talk that we had has just today been postponed or canceled. We're looking for an alternative talk. I will keep everybody posted about that.
Okay, thank you very much. Bye bye.